Welcome everyone to the Global Soil Biodiversity Initiative webinar, GSBI Speaks. We've got an exciting topic for you this morning. We're really glad to have you. And you might want to just sign up to receive the latest news, blogs, and highlights on our website. This morning, we want to also pay attention to the assessment report that just came out in December. That's the next slide, I think will be coming along. And that shows 300 plus scientists were committed to writing an assessment of the status of soil biodiversity and what are the potentials for soil biodiversity and management. So if you haven't checked that out, please do. I'd also like to mention some housekeeping things before we get started. First of all, this is being recorded. Secondly, there's some questions. Please put your questions and answers in the question and answer box, but the panelist will not be writing answers back to you. And so we will try to answer. Wim van der Putten is going to moderate this, and Wim will try to answer all the questions or give them to panelists. And I think that's the most effective way that we'll be going. So with that, I'd like to introduce Wim van der Putten from NIOO at the Netherlands, Wageningen. Wim?
Okay, so there we go. So I see 287 participants already there, and still people are just walking in. So it's my great pleasure to introduce the panel to you. And the first one I'm going to introduce is Tancredi Caruso. He's from University College in Dublin. And Tancredi, his work focuses on processes that structure microbial and animal biodiversity, and he's interested in processes in both space and time. And his work is particularly interesting because he takes a very theoretical entry into the topic, and that's also why he is the first speaker. So we start with some more theoretical background. Tancredi has traveled all over Europe. He has received funds and grants from all kinds of different countries and agencies. I didn't even know they would exist, but he also got an EU Marie Curie grant, which is quite prestigious to get at in Europe. And currently he's working in Dublin. So the second person who is going to speak is Cameron Wagg. And Cameron now is in Canada. He's a research scientist in agroecology, biodiversity and ecosystem functioning, and he works at Agriculture and Agri-Food in Canada. But he also has been traveling around quite a bit. So he has been in Switzerland, where he did his famous paper on multifunctionality of soil biodiversity in 2014. But he also worked in the Jena experiment. And currently he is back, or he's in Canada, where he works on effects of extreme weather events on crop plant performance and productivity. So he brings in the agroecological component of today. And this is quite important because he's studying how crops perform under extreme drought. Then the third speaker, I'm very glad that Mary Firestone is on board here. And she's at the University of California, Berkeley. Actually, I think almost all the occasions that I've met Mary was in China for one reason or another. I don't know why. But she's a soil microbiologist, and she's particularly interested in microbial processing of carbon and nitrogen in soil and how this influences the capacity of soil to support plant growth in agriculture, but also in rangeland, forests, and wetlands, so everywhere where you can think of. And Mary is world famous for her studies on a wide variety of subjects. And she's a leader in the field of plant soil systems responses to drought and also rewetting. And she has a clear eye for considering heterogeneity in soil. And some people see that as a pain, but she looks at it as a challenge. And actually, she also doesn't hesitate to take advanced technology in her studies, like metagenomics, stable isotope probing, nanosims. And I think that her work is really contributing to understanding terrestrial ecosystem responses to global change and many other factors. And then last but not least, we have Franciska de Vries. She is currently based at the University of Amsterdam, where she is professor in, now I have to say it correctly, in Earth and Surface Science, which is quite a broad area. So she arrived at Amsterdam starting in Wageningen for her PhD thesis on effects of on interactions between soil fungi and nutrient cycling. And then she moved to the University of Lancaster, and there she worked on 2 EU projects named Soil Servers and Ecofinders. And then she got personal grants from, for example, BBSRC. She moved to Manchester, and she developed her own research group there as a senior research fellow in the end. And from there, she went to Amsterdam. Franciska wrote a very interesting paper on harnessing plants against drought by using microbes. That was published in Science some time ago. And I think with all these speakers, we have a great group of people who can introduce, they will introduce the subject to you, and I hope they will raise a lot of questions that we can see in the chat. So this is my introduction to the speakers. And then I'm going to share my screen for some slides of introduction. And can you all see my screen? Yeah, I see some people nodding. So the topic of today will be soil biodiversity and resilience to climate extremes. So why is this so important? Well, these are the speakers. If you look at the incidence of extreme climate events since 1980, then what you see is that actually many of these extreme events are increasing. So we have increasing hydrological events. We have like flood and mass movement, so that means landslides and so on. We get an increase in storms, an increase in extreme temperature, drought and fires, and a slight increase in geophysical events, earthquakes, tsunamis, and volcano eruptions. So it seems that going along with climate change, that we also get an increase of these extreme weather events. And these extreme events have a massive effect on the functioning of soils and soil biodiversity. So this graph comes from a report from the ESAC, which is the European Academies of Science Advisory Council. It's interesting because what they did not plot in this graph is, for example, the number of extreme droughts in my part of Europe, which is the northwestern part of Europe. And actually we had a severe drought in 1976, and which was then the first drought in many decades. And then we didn't have a drought for a long time, but then we got droughts in 2003, 2006, and then over the past 3 years, we had 3 dry summers in a row. So this clearly shows that the number of dry summers is rapidly increasing, and it seems that this is just very much related to the current climate change, climate warming. So, so that's one part of the title of this meeting, extreme events. The second aspect is soil biodiversity. So soil biodiversity is, I think, what many of us is binding. When you speak with most people and also with farmers, they say, well, soil biodiversity is earthworms. Well, soil biodiversity is much more than that. So we have, as you all will know or may know, if you have one handful of soil, you may have 5,000 taxa of microbes and invertebrates in your hands. So you have a whole zoo, or even more than a zoo, almost a tropical rainforest of biodiversity in your hands. including viruses and bacteria and fungi and nematodes and amphitraids and microarthropods, earthworms, and whatever you can imagine. So, so if we think about biodiversity, we think about more than only one group of species. Actually, we think about all species that can be present in soil. So what is happening with soil biodiversity currently? So that was nicely worked out by Stefan Geissen in the paper where he involved me and Diana Wall in. And he came up with some conceptual graphs showing or arguing that with increasing human impact, that you get a decline of soil biodiversity. This has been demonstrated in several studies, and also that leads to a decline in multifunctionality. But what you get with in soils that are more under human pressure, you get an increase of incidence of pathogens, but also an increase of management dependency. And you get an increase of pollination and all kinds of pollution and all kinds of side effects. So this all leads to a decline of sustainability and an increase of costs. And maybe this discussion is playing in more parts of the world, but here in the Netherlands, people sometimes start attacking ecologists or soil ecologists when we explain that it's going bad with, it's going worse with soils. And then they say, why is it going bad with soils? Because we have the highest productivity worldwide almost. And then we have to explain, well, what you do in agricultural soils is you're sort of specializing on getting high productivity, but this goes at the expense of all other ecosystem services and functions provided by the soil biodiversity. And so this is sort of the mind switch that we need to try to bring to people so that they understand, well, you can be very effective or very, you can have a very good result, for example, in getting agriculture production, but if all the other functions are sort of declining, then the question is, who is going to pay for this? Okay, so that's a little bit about soil biodiversity, but soil biodiversity is not only something about species, it's also about how species are interacting with each other. And so I think about 2 decades ago and following, there were a number of studies linking below ground to above ground interactions. And this is one of these cartoons showing that actually what is happening in soil is very much dependent on each other. So if plants are attacked by pathogens or if they are aided by symbionts, that also affects the way how plants may deal with the decomposers, for example, and with other soil organisms. But also that affects how plants deal with above ground biota. And so ultimately, if we think about if we think about soil biodiversity, we have to think about soil biodiversity in full combination. So the soil organisms that are linked to each other in food webs. And so you can't just look at soil biodiversity as 1, 2, 3, 4, 8, 16 species or so. You have to look at it from the perspective of the roles of these species in the soil food web. Okay, and then the third aspect is the resilience aspect. So what does resilience mean? Actually, resilience means that it's like if you just stretch a spring, then if you let it just go, it will come back to the same shape. And so the question is actually what will happen under extreme weather events if you look at soil biodiversity in its full context. Do we get the same system back after the extreme event or not? And that is a topic that I hope that the 4 speakers will also touch upon and that we can discuss. So those are my words of introduction, and I think I will now quickly hand over the word to the first speaker, who is Tancredi Caruso. Tancredi, the floor is yours. If you can unmute.
Thank you for the introduction. I'm going to share my screen to put up my slides. Okay, so again, thank you for the introduction. Let me just go straight to the presentation. The relationship between soil biodiversity and climate extreme is a complex relationship. That's because soil ecological networks are controlled by many factors, including plants and interactions between soil organisms and plants, but also environmental variation, the chemical and physical properties of soils, and of course, perturbations. And all these things interact to affect processes such as production, fluxes of energy, matter, etc. The structure of ecological networks in soil control the expression of the functions. And this structure is, however, very variable over time, and so the functions vary over time. Whenever perturbation, there are changes in the structure of the network, and that implies changes in the functions that the communities express. And after a certain amount of time, when the perturbation is released, you might have some recovery. And there are many possibilities. One possibility is that the network returns to its original state, and so the functions will follow. Or the network could go into another state or other states. And there are very many questions. How many states? Are the states stable? What does it mean that the network structure changes? Is it the connections between different species that changes, or is it the intensity in the connections, or it's both? Now, these are questions that are difficult to answer, especially nowadays, because when we look at climate streams, we mostly focus nowadays on multiple perturbations. And when it comes to multiple perturbation, you need to ask very many questions, such as whether, for example, the nature, intensity, and order of perturbation matter in terms of the response of the community to the perturbation. So, for example, if you have drought and then flood, doesn't matter whether you have first drought and then flood, or maybe first you have flood and drought. Is the response different? And so that's a difficult question to answer. There are also questions in terms of how often we have to sample the communities, and when. Do we, for example, have to sample in between perturbations, or we are just happy with before the perturbation and after the perturbation regime? And if you look at this figure, yeah, each of this panel is one particular community. But of course, we need to replicate those over space. And that means that we're going to do these in many locations, possibly over a large landscape, or even at the scale of the globe. And so we're going to have multiple perturbation and control systems, but the landscape itself, it's very variable. It's variable surely over space, but also in time. So there are big challenges. If certain conditions are met, some of these challenges can be actually resolved. And this is where I'm conducting some research at the moment. And in particular, let's assume that if you have a community which is not perturbed, so a control community, it's relatively stable over time. There will be fluctuations, but it will be roughly stable over time. And so you have multiple communities, rep 1, rep 2, maybe rep n, a number of communities. And for each community, you can measure a particular property. I don't know, species richness in the community, or maybe even the activity of an enzyme, any property you want to measure. And so there will be some fluctuations over time in this property. So there will be some variation over time. So if the variation over time in a single community is comparable to the variation in space across the communities, we can simplify our experiments, because basically we don't need to invest too much in the the replication over time of the before part. We can mostly concentrate on the aftermath of the perturbation regime. And that would simplify things a lot. But when you are back to Earth, things might not be so simple. And this is because of the extreme variability you observe in soil, spatially and over time. And this is my conclusive slide where I want to demonstrate that. And it's a simple experiment. It's a very popular experiment nowadays. It's a rain shelter experiment where you can basically create a severe spell of drought using this rain shelter. And here you have the control plot. And in this case, you can see a simple property, soil moisture, measured over a number of days after we remove the shelters. And you can see that surely after the removal of the shelter, drought clearly affected soil moisture. So you have much less water in the soil in the drought than in the control. But what is really interesting in this figure, it's what's happening to the control. The control is extremely variable, and actually there's a change of regime in the control, which is violating actually what I said before in terms of a relatively stable control. And these peaks here are pockets of rain. And you can see that the drought is picking up the water from the pockets of rain just pretty only 2 weeks, or maybe actually 3 weeks after we remove the perturbation. And so after that, you can see how the fluctuations in the recovery are similar to what you observe in the control. And there are very many lessons to learn from this picture. We learn that recovery after a severe spell of drought, in this case, it's not a linear process. It consists of trends, for sure, there is a recovery, but it also consists of periodic fluctuations and stochastic fluctuations. And it takes time to recover, even when conditions become favorable. And I suppose that what I would like to see in the future is this kind of data with this kind of time variation, also for soil biodiversity data, for ecological networks, and maybe also for functions. And with that, I will just stop it there, hand over back to Wim.
Thank you, Tancredi, for pointing out that actually, if you look at resilience, then the question is, what is resilience? What are you looking at? So we will just keep the discussion for at the end after all the presentations. So I would like to ask Cameron Wagg to show up and give your introduction. And Tancredi, you can close your video and mute your microphone.
Okay. Okay, here we go. So a big thanks to Wim and Diana for the invitation here. It's a real privilege to be on this panel and share some of the research that I'm doing. So my research largely focuses on how soil biodiversity functions. So whether it's increasing the number of arbuscular mycorrhizal fungi in the soils, these are plant symbiotic fungi, we can increase the productivity of the plant. But also if we have more microbes and invertebrates in our soil, the more diverse soils we have, we can also enhance multiple functions. So things like related to nutrient cycling processes. If we have a greater soil biodiversity, we can reduce nutrient losses through leaching, N2O emissions, which is a pretty key important greenhouse gas. We can enhance litter decomposition, nutrient cycling that then the plants can take up. And so this is because if we have more diversity in our soils, we have more of these complex networks and associations that we just heard about that together work as a consortia to promote some of these ecosystem functions. So for example, litter decomposition is not done by a single bacteria or a single fungi. It's a bunch of different fungi and bacteria working in consortia to complete the process through time. So these diverse soil communities can be quite important for ecosystem sustainability or the stability of an ecosystem. So this is, will come out next week in eLife, but if we have soil that's basically devoid of soil biodiversity here in orange, and then we have a little bit more soil biodiversity in yellow, green, and our full soil biodiversity that we can have in our grassland fields, we can increase the productivity. But this isn't very stable over time. As time progresses, in the absence of most soil biodiversity, we have this boom in productivity because the grasses are uninhibited by competition and perhaps pathogens, and they're sucking up all the nutrients. But this crashes quite heavily over time. And this is because we don't have these key soil organisms and functional groups in our soil to maintain these ecosystem processes. So we're not fixing nitrogen, litter decomposition is breaking down, and we're losing nutrients from the system, and this destabilizes the productivity. So soil biodiversity is quite important for a number of ecosystem functions and sustaining productivity through time. So what about in the real world? This is a nice paper by Gina Garland that came out recently in Nature Food. I was really privileged to be able to help her out with some of the synthesis. So this data is from a Biodiversity project that includes 5 different countries and a number of different researchers. But what you're really seeing here is the complexity of the environmental gradient from southern Spain up to more northern Sweden on soil microbial diversity. So mean annual temperature and precipitation can have strong effects on the structure and the diversity of the soil microbial community. And this can have an effect on yield in agricultural fields. So for example, in hotter, drier regions, this drier temperature might reduce bacterial diversity that will play into the ability to produce higher yields. But what's interesting is that we can also manage to perhaps buffer against some of these climate effects. So for example, longer crop cover throughout the year, we can enhance bacterial diversity, and perhaps having a greater diverse cropping system, we can enhance some of these microbial diversity properties that are going to sustain nutrient cycling, and that's going to eventually play into sustainable yields. A project that is still going on and in the midst, this is led by Louis Pierre and Claudia Goye. So we're doing the same thing, but over in Canada here on the East Coast. So we're not just looking at sequencing the soils and the microbes, but we're also looking at bacteria, anthropods, nematodes, mycorrhiza, soil enzymes, and soil organic matter fractions across a large environmental gradient. So these red dots are all the sampling points. And from this data, we're going to hopefully be able to identify cold and hot spots of biodiversity and associated ecosystem functions and how that might change across extreme climate gradients here in Eastern Canada. So stay tuned. Hopefully more will come out of this in future years. But I've also worked quite a bit on plant biodiversity with the yen experiment. And I thought I'd share this just quickly to sort of illustrate the importance of diversity, especially in perhaps plant communities are a way that we can use to buffer against climate extremes. So this is a rainout shelter experiment from the yen experiment. And red is when we impose a summer drought, and blue is the control. So if you don't water plants, when you have 16 species, high plant diversity, we still lose some productivity, but it comes back in the spring. And we've seen this over a 7-year period. So we have, we lose a little bit of productivity, but we have this really high resistance, or we call it compensatory recovery. So the long-term means in the productivity hasn't really changed. But that doesn't really happen when we have a single plant species. For example, in a monoculture in an agricultural field, we lose productivity through the drought and it doesn't recover very well. So diversity can be important for this recovery process. So putting this all into kind of a summary, we know our climate is changing and we're having these extreme climate events and drought years, as Wim was mentioning. But I think we might be able to overcome this and promote soil biodiversity for sustainable ecosystem functioning with diverse plant communities. So this is a project that, or concepts that I'm quite interested in, that relates to regenerative agriculture. And at the moment, we're screening all kinds of different crops and diverse mixtures of crops to see how they promote soil biodiversity and ecosystem functioning. And this is really the concept of regenerative agriculture. This is an infographic from General Mills. But if you look at what they're saying here, minimizing soil disturbance, keeping the soil covered, maintaining year-round living roots, and diverse cropping systems, these are all practices to create conditions to enhance soil biodiversity and maintain our soils for future sustainability in agricultural systems. That's all I have for you today, so I'll hand it back to Wim.
Okay, thank you, Cameron. Very nice. We bring regenerative agriculture into picture. So Mary, what is the points that you would like to bring in?
I'm going to need the button to share my screen. There it is. Okay, I also would like to thank Diana and William for the invitation to talk about a little bit about our work, but mostly about water in soil and how it controls life. So most of the work we've done for the past 20 to 30 years has been centered in California. And California, most of California is a semi-arid ecosystem. So a lot of the work we've done on water is based in a Mediterranean type climate, a semi-arid ecosystem grassland, annual grassland in California. And we've worked a lot on life history strategies in response to drought, life history strategies of the organisms in soil, starting many years ago with bacteria, but more over the past 10 years, including fungi, fauna, and viruses in our work. And so we've looked at response to dry down and response to wet up, how soil microorganisms tolerate that. And we start with the fundamentals, and that is, what does it look like if you're sitting in soil and you're a microbe? And what you see is the water you're living in. And as the soil dries out, the water films that you experience become thin very quickly, such that by the time you get to about minus 15 bars, you have about a 10 micron thick, no, 10 water molecules on the surfaces that you're trying to live on. So water film thickness decreases rapidly, and limitation of motility, particularly of the water-based fauna, occurs at relatively high water contents pretty quickly after a rainfall event. So diffusional limitation occurs, and then the water films, the thickness of the water films, as it controls the supply of nutrient compounds, begins to limit nutrient availability to organisms in soil as well as to plants. But both microbes and plants have some control on the water content in their immediate habitat. Microbes in particular can control the water content near their homes, if you will, by producing extra exopolysaccharide matrices or extra polymeric substances. And these are sponges, if you will, that are extremely effective at holding water. Plant roots also produce the slimy viscous substance around the root tip, or mucigel, which serves to retain water and also bridge gaps between the root and surrounding soil. So the reason this material is important is that in water potential equilibrium, EPS retains more water, has a higher theta or water content than does the surrounding soil. It cannot change the energetic availability of the water. But the higher water content in the EPS allows diffusional supply to continue even at lower energetic availability or lower metric potentials. EPS also slows the rate of change of water content and water potential in the microhabitat, thus giving organisms potentially a little longer to physiologically adapt to desiccation or physiologically adapt to the wet up. So 30 years worth of work we've worked on what happens as soil dries. First, you tend to inhibit the motility, starting with the largest organisms, but we've also gone all the way to viruses, to phage, in terms of how soil water content restricts the mobility of phage. As the soil dries, you begin to limit diffusional supply, and eventually the response is one of metabolic impairment or a response such as spore formation if the organism has the capacity. So dry soil, water controls substrate and nutrient availability to microbes and plants, as well as the physiological state of both the microbes and plants. But both microbes and roots have some control of that water content. I'm going to talk briefly about, we talked about EPS, extracellular polysaccharide production, which can provide pathways for water and nutrients to move to roots and potentially bridge water film discontinuities or gaps. We've also done quite a bit of work on AMF and the capacity of arbuscular mycorrhizal fungi to transport a substantial portion of a plant's water demand. And we have found in our studies that the water is moving outside of the fungal membrane, presumably outside the wall or in the wall. And this water can carry nutrients as it is free water on the outside of fungal hyphae, and also has capacity to bridge water film discontinuities. So what do we need to understand? A great deal, clearly, but the points that we are very interested in, the differential effects of drought on the different groups of soil organisms. Mesofauna that live effectively in the soil air, microfauna, the organisms that function in water, fungi, bacteria, and viruses. And the water effects on each of these groups can be profoundly different. We are asking whether we are able to enhance water retention and transport pathways in soil. And part of that is to understand the role played by soil microbial community in mitigating plant response to drought and assess management strategies that use the soil biota to enhance plant drought tolerance. And I wrote this this is the introduction to Franciska's talk, who is the next speaker.
Thank you, Mary. Yeah, a better bridge. I could not do that, and you did it to Franciska. And I think quite some questions will remain. So what is the evolutionary benefit for the microbes? But let's talk about that in the discussion later on. Franciska, you have a bridge, so yeah, please cross it.
Right. Thanks very much. Thanks also again to the organizers for giving us the opportunity to show you some exciting research. So yeah, Mary has, as she said, given the perfect introduction to what I'm going to talk about, because when we look at soils, we've talked about the benefits of soil life, soil microbes for plants. They mineralize nutrients. But when we look at the soil, most soils on Earth actually have plants growing in them. And these plants, they actually influence the microbes as well. So this is a two-way interaction. It's not just that the microbes perform functions that are important for plant growth, but the plants also fuel the microbes. So when we zoom in on these roots, really these roots, they interact in many, many ways with, in particular, the microbes that live in the soil. So roots can take up water, they can take up nutrients, but roots and microbes, they also compete for nutrients because they both need nutrients. As has already been mentioned, microbes, they decompose organic matter and they mineralize nutrients, but roots also directly collaborate with microbes, for example, with mycorrhizal fungi, but also with nitrogen fixing bacteria. But importantly, roots also exude substances into the soil that contain carbon, sugars, but also signaling molecules. And so these root exudates are actually a means of communicating between roots and microbes, so they can fuel microbes with their carbon, but they can also attract them and deter microbes, for example, microbes that attack plant roots. So there are really a lot of ways through which roots and microbes interact. And these are important if we want to understand the response of an ecosystem to drought. Because global change, it obviously can directly affect soil biodiversity and soil organisms through, for example, its effects on that water film that Mary just said. But it can also indirectly affect what is happening in the soil, both through affecting how individual plants perform and function, but also through changing plant communities and through affecting these plants and their roots. It can affect the interactions between roots and microbes, and these interactions are really crucial for the processes in the soil, such as carbon storage, but also nutrient recycling. So what we know is that drought affects different plant species differently, but we actually know surprisingly little about what sort of predicts the response of different plants to drought. And this is just to show you that different functional groups also respond differently to drought. And on the left, you can actually see the very first results of a meta-analysis that we're currently working on. This is my postdoc, Mariana Gleish-Silva, who's leading this. And this shows that overall, legumes are actually more affected by drought, more reduced in biomass than grasses and forbs, for example. But in response, that's in response to a drought. But what is really interesting is actually what is happening after a drought is ended, and then actually those changes might completely shift. So on the right, for example, is just a structure equation model that we did on the response of plant communities to drought. And what we found there was actually that this fast-growing grass, Dactylus glomerata or cocksfoot, was initially very strongly reduced by drought in a community. But when the drought ended, this species really took over in the communities, and this increase in abundance of this exploitative grass continued to affect the soil and the soil organisms, the bacterial and the fungal communities. So how drought affects different plant species can actually affect what's going on below ground, and this can have implications for the functioning of the ecosystem. And just to show you that, this is a sampling that we did in a long-term drought experiment in the Netherlands, in a Dutch heathland. And this is an experiment that's experienced 15 years of summer drought. And what we did is we sampled underneath 2 functional groups of plants that occur in these plots, in drought plots and in control plots. So this is heather plants, so that's a shrub, Calluna vulgaris, and also the grasses that occur, which is mainly the Shamsia flexuosa. So both these species occur in this system, but what we found is that in response to that really long-term drought, that the soils underneath the grasses, which you can see here for drought and control in blue, and drought in orange, you can see that the soil underneath these grass individuals had lost carbon in response to that drought, and also the microbial biomass was reduced more under the grass than under the heather. So this really has implications for ecosystem functioning, because if the abundance of these species shifts, then that might actually make the system more vulnerable to drought, and the ecosystem might start losing carbon under these recurring droughts. And we think that this might also be because of how different plants exude carbon into the soil, and how drought or other climate extremes can affect these carbon inputs through root exudation into the soil. And these are some very recent results from my postdoc, Alex Williams. And this is just on the left. You can actually see for 2 different species. So that's Anthoxanthum odoratum, which is sweet vernal grass, and Dactylus glomerata in green, that they differ very strongly in their root exudates, but also that these root exudates are very strongly affected by drought, but in particular by warming. So really the whole exudate profile shifts when you have warming, but also when you have drought and warming combined. So the composition of these exudates that enter the soil changes, but also, as you can see here, the amount can change in response to drought and warming. And in a previous experiment, we actually found that these drought-induced changes in what these plants exude into the soil have implications for the functioning of the soil. And in this case, exudates from plants that had experienced drought actually increased soil respiration. So they might increase the loss of carbon from soil in response to a drought. And so this is just really a very brief snapshot of what I wanted to show you, that we really need to be looking at these interactions between roots and microbes if we want to understand ecosystem response to climate change, and also really to highlight that there's a lot of things that we don't know yet. So what's the role of the root microbiome in plant response to drought? And what are the mechanisms through which the plants recruits these maybe beneficial microbes that might help them survive the drought? And what are the components of root exudates that change in response to drought and warming? And yeah, there are a lot of questions that we still need to answer. And one of the things that makes that actually quite difficult is that soil biodiversity is obviously very difficult to look at, but I would say that root exudates are they're even harder. So sometimes I curse myself that I've sort of entered this direction. So that's really what I wanted to show you.
Thank you very much, Franciska. Yeah, I can imagine that you curse yourself about measuring root exudates because that's a hell of a job. So actually you brought back the point very nicely to the start made by Tancredi. So what to measure, how to measure. Maybe all the speakers can now turn the video on and their microphone. So we have, as far as I can see, but maybe there are more, over 400 participants now. And we have already a number of questions in the Q&A. So if you have a question, don't raise your hand because that function is not working. Put it in the Q&A and then we go through the Q&A and I'll try to sort them a little bit to topic. So some of the first questions that I see are about how to measure and how to deal with this spatial and temporal dimensions that Tancredi put on the map. And maybe I could ask each of you your view on, from your perspective, so what to measure and when and how to measure if you want to study effects of extreme weather events on soil biodiversity and ecosystem resilience. So maybe Tancredi, can you just start?
Yes. The first thing I like to say is that in the past, there has been an emphasis on spatial replication. And we don't have so much information on variation of communities and response to perturbations over time, especially in the field. In glasshouse experiments and laboratory experiments, it is kind of more, it's easier and more natural to have multiple sampling over time. But in the field, we don't have very much information on the variation of these communities over time. And I think that is critical because regardless of whether variation in space or in time is bigger or smaller, for example, for microbes, it might be that actually variation over space is bigger than variation over time. But despite that, if we speak of resilience and if we speak of recovery, automatically we are entering the time dimension. And so First of all, it's important possibly that there's a shift towards studies that have more investment into the temporal dimension of this community. That's not easy, because very often our grants don't last very long. And so, you know, setting up an experiments and investing time and resources over time, and then maybe something fail, it can be, you know, a big loss. But scientifically, I think this is an important aspect. We need to invest more, in my opinion, in temporal replication. And then there's the second one, and then automatically, then you ask yourself, when shall I sample? And that's not an easy, again, sure, it's not an easy question. And I will never say when to sample in general terms, because in general terms, this question doesn't make, you know, it is really, it depends on the questions, it depends on the system, it depends on the organisms, it depends on so many things that it is a very interesting question, but it tends to be very system dependent, the question to when to sample. So just leave it there for the moment.
Okay, let's bring it to Cameron. What would you say?
Yeah, that's a good question. I would kind of have a similar answer. It depends on what the organisms are and what the ecosystem function of interest is. So from an applied setting, if you're a farmer, for example, it might be yield, and that's one measurement that at one specific time. But if we want to associate that to soil biodiversity, which can be quite dynamic on short timescales, the more we can sample throughout, for example, the growing season, the better. But I think, as we all know, that sort of kind of would be really great to have, but to go and sample and sequence, and it's quite a lot of work and expensive to do. So we're kind of limited by a few time points. So I would, yeah, kind of echo the effect that it would be great to have more resolution on these temporal dynamics in soil communities under real world settings, to see how that dynamics play into the resilience of the ecosystem and the productivity of the ecosystem.
Mary, you are not hampered by measurement capacity, so what would you do?
Well, I'd like to go back to something that was said before, and that is the importance of long-term measurements, particularly if you're looking at impact of drought. Extreme events, yes, for sure, but also the long-term changes. Unfortunately, when you go long, it becomes very difficult to get intensive measurements, for sure. So that requires choices of what you will measure and how frequently. As Cameron said, one of the things that's critical is the plant productivity. Usually we are after something to do with either crops or fiber production. But I'd also suggest that the other critical properties or products, if you will, of soil are carbon stabilization or carbon sequestration and potentially trace gases. These are probably three of the primary reasons we're interested in biodiversity. So changes in soil organic carbon happen slowly over a long term. Franciska's been able to see them over 15 years. We've done very similar experiments over a period of 2 years and see very little effect, because I think 2 years, unfortunately, is not long enough to see impacts that on the functioning, the critical functioning of the ecosystem. So perhaps set up studies that have more intensively sampled plots, and then plots that go long, that will allow you to look at the long-term impacts.
Okay, Franciska.
Yeah, well, I echo actually everything that's already been said, but I just want to add one thing. Because actually in the past, when people looked at disturbances like drought, for example, a lot of these studies actually end when the drought ends. And sometimes they go on for a week or maybe a month. But actually this sort of recovery trajectory, I think that is actually one of the most interesting things when you look at these disturbances. And that's actually when the fast sequence of things happen that will determine in which direction the system is going. So I really think that if we want to understand better how our ecosystems respond to these extreme events, I think we need to sample beyond those events, and also, well, as Tancredi showed, also look at multiple events and look at the recovery and what the implications are of a previous disturbance to the response of that system to another disturbance.
Okay, so it's very important to define your question, but also to think carefully about the system. In some cases, you might look at biomass as a proxy for ecosystem functioning, but in other cases, it's more carbon storage or so. And also the point is that we need long-term studies, and also we need to look beyond the effects of one disturbance. So not stop after the disturbance is restored, but then the most interesting part may start. That actually brings me to the question, so what is resilience? And maybe we can just go back in the reverse order, because Franciska sort of mentioned that already. So Franciska, what would you consider resilience of soil biodiversity?
Well, I would generally consider resilience as something that is disturbed and then moves back into the direction that it was, and also continues functioning like it was. And I guess actually I guess I'm particularly interested actually in how the communities change, but I think when we talk about resilience, many people are talking about the functioning of the system, but I would really like to include not just the functioning, but also the form or the community of the system.
So Mary, do you have an impression of whether the communities can become restored completely as they were before, or do communities of microbes, for example, become exactly restored, or do you always get something which is slightly different? How does it work in your case?
So that kind of goes back to some of the basic literature on resilience, and that is resilience doesn't necessitate restoring the starting point, but rather restoring a functional, that meets your functional needs. So with microbes, particularly after extreme events, you may have changed the composition of the community. But what we care about is whether we reestablished the functioning and the critical functionalities and the interactions. So rather than simply looking at a change in composition, I think we need to think about what we want from resilience, how we want the ecosystem.
Okay, so that brings me to the functional person here, Cameron. So how about restoring multifunctionality? Do you have any examples that multifunctionality can be lost because of extreme events?
You've got me thinking off the top of my head, scratching my head, but I think it's a really good point that, the long-term studies, for example, the drought experiment that I showed in the yen experiment, we didn't really see these long-term patterns after a few years. We might have thought, oh, it's a fluke that we have this high recovery or compensatory recovery, but we see that it's a real thing after we kept the experiment running for nearly a decade. For resilience, I think for me, it sort of encompasses both the resistance and recovery parts. So an ecosystem that is highly resilient might also have high resistance. So it won't be as negatively influenced by a climate perturbation, as well as this more rapid recovery in the functioning. So I guess that's what I would have to say. I think some of the open questions, at least from my side too, are also like what Franciska was mentioning. What happens to the composition of these soil communities as they recover, and how long does it take them to recover to restore the soil, the functioning of the soil that we depend upon?
So I want to slightly adapt the question, because I just see a comment of Katrina Hedlund that many of the soil organisms, especially microbes, just sit and wait for better times. So Tancredi, from your perspective, what would you do with these sitters and waiters? So with the silent majority that is just hanging around and needs to be what Patrick Lavelle would call to be kissed alive at some stage. So how do you handle that in your work?
I find this very fascinating, the fact that very different players can actually return the same functions to you, which is something very microbial in nature, but I think it's really a general aspect of soil biodiversity. And so this is really essential. And the thing there, what is important is the structure of the interactions in the network. It doesn't matter who are the players, the species or the particular organisms, as long as you have the right structure in place, the functions will be delivered. So a critical question is whether the perturbation is going to basically be so detrimental that you may lose the minimum number of players that are necessary to get that particular structure. And I would say, Jay, we don't know. In many experiments, it seems the community is quite capable of returning to certain level of functions. Maybe there will be changes in the composition of the communities, but overall, you may observe a quite remarkable ability of many soil systems to return to a certain level of functions. So, yeah, we don't need to know the nitty-gritty details of every species and every aspect of the community. What is important is that those particular interactions are in place. And so this is where methodologies like network analysis or more modeling approach can be very useful. Another thing I'd like to say about this is that resilience, I mean, the others, Franciska, Mary, and Cameron have touched on the most important aspect in terms of the ecology and biology. One thing I'd like to add is that resilience is a property that has to do with the velocity at which the system recovers. So again, there is an important dimension in terms of time. And two systems may be able to recover to the original state, but at very different velocity. And the velocity is also important. How quickly or how slowly does the system recover? And this can be critical. It can be critical for application. It can be critical for our ability to produce food. So there is this element of velocity and rate in resilience, which I think it's particularly interesting.
It's interesting, this point about velocity, because so we had an experiment here done by Marta Marubia, and she looked at Mediterranean sort of or Eastern or continental European soil communities versus Northwestern European soil communities and how they recovered after drought. And actually we got something completely opposite to what at least I had expected in the beginning. So I would have expected that Mediterranean or continental soil communities would recover much stronger because they're used to extreme drought. But they did not do so. The most strongest responders were the Northwestern soil communities, Northwestern European soil communities, so the ones more from the temperate, more rainy environments. And then we started thinking about that, and in the end we thought maybe this is not unlogic, because these continental soil communities have evolved in a system where one drought could always be succeeded by another drought. So the first rainfall may not be the start of a full rainfall season, and therefore it's much smarter for them to just sit and wait for longer until there is enough rain falling, and instead of just doing like the stupid Dutch microbes were doing, immediately when there was rain, they started to become active. So this velocity and also response rate, and so that would be something of interest, perhaps. I'm at the same time, I'm browsing through the questions and I'm trying to group questions, and I see a question from Beatrice Guerra, and she is asking something about new adaptive or mutant species in the face of climate change. So maybe Mary, could you say something about that? And maybe in relation to this, could you also say something about what is the benefit for microbes to make these polysaccharides? So why would they do that?
Okay. So how quickly can microbes adapt to changing conditions? And so the traditional ecosystem perspective has been that stress on bacteria doesn't matter because they evolve so quickly, but they don't. They don't necessarily evolve as quickly as people would expect them to. There is a huge reservoir of genetic diversity, and that is usually what buffers the functionality of a system, I would say. But there is also clearly the capacity, the genetic variety within a population even would allow, whether it's mutation or not, I don't know. Why do microbes make EPS? I look at the EPS matrices that microbes live in as kind of the little house that they've constructed to live in. They are able to produce extracellular enzymes that are contained within a goo. So the goo becomes a digestive goo and is able to encompass particulate and larger pieces of substrate, beginning to digest it. It retains the product in a close by state. The EPS is a water holding, so it gives you buffering in terms of wetting and drying and water content. In some cases, it protects from predation, although in other cases, there are protozoa that specialize in grazing bacteria that live in EPS. So that can go both ways. The expectation of people who work in this area is that most bacteria and likely fungi produce some to a lot of EPS when grown in a soil matrix.
So Franciska, how would you consider the benefit of the different partners in your plant microbe interactions under drought? So if plants are not protected by microbes, so what's the benefit of one and the other player in the system?
Well, clearly plants, if they want to collaborate with these beneficial microbes, they need to also put in something. And so I think how I think it works is that plants that invest in these positive collaborations with microbes, for example, AMF or EMF, that those plants, they are probably generally actually more resistant to the drought, so better able to cope with the drought, whereas these more faster growing plants that are actually more vulnerable to the drought, they tend to be they tend to not be very careful with their carbon, so they might exude a lot of carbon into the soil and actually attract maybe the microbes that are also really quick to grow and to then maybe mineralize the nutrients for the plants to maybe recover after the drought. So that's sort of how I would hypothesize sort of the investment of the plant into the microbes.
So is this on purpose or is it just by accident? So I want to draw the comparison with above ground plant defenses through defensive metabolites or that are induced based on herbivory or so, or green leaf volatiles. Is that there's also quite some discussion there whether this is on purpose or whether it's just an accident. So it's a response to something else and then everything responds to that. What do you think about this?
Well, I think people always used to think that root exudation was just a passive process and a loss of carbon to the plant, but I think it makes much more sense to assume that it's actually a plant strategy and to see that collaboration of the plant with the microbes as like the part of the plant, just as we see now that sort of the gut microbiome is actually something that benefits your own health if it's healthy. And I think we can sort of extend the plant to its microbiome, really, not just in the soil, but also on the leaf. And that's not something that I know a lot about, but there's similar interactions actually going on the surface of the leaf and inside the leaf. So I think this is not all accidental.
Yeah, so grasses that have endophytes are more drought tolerant than grasses that don't have endophytes, for example. So I also see some questions also from Hartmut Köhler and others about soil animals. So it's now a lot about soil microbes. So let's just start asking Cameron and then Tancredi about, so if you look at the other components in the soil food web, so beyond microbes, so how do they play a role in this soil biodiversity responses to drought and the resilience to it? So Cameron first.
So I don't have a lot of experience with macro and meso soil fauna, but we have bacterial feeders and fungal feeding nematodes. So there's a whole trophic cascade of interactions that are going on in some of these feeding food web networks in the soil. So they are definitely quite important for regulating a lot of how these soil microbes function. I haven't done a lot of work on it myself, but that's a great question, because if we start removing, some of these top soil carnivores, or however we want to call them, that's going to change the whole dynamic and structure of the soil food web and how it functions.
So Tancredi, what would be your ideas about this?
So we used to think that plant microbes would be more important and animals would be kind of more collateral. But very recent stable isotope studies show that much of the carbon that is going from the roots of the microbes eventually also ends up into the animals. So they're actually very tightly interacting also with all the microbes in the rhizosphere. Sure, I'm speaking of the smaller animals, but also the relatively larger one, the mesofauna. So not just the nematodes, but also things like very small arthropods, collembolans, mites. Some of the carbon they have, it's coming from the root exudates via the microbes. Most likely, we don't really know. It could be also that it goes into the organic matter, then it goes into the bacteria, and then the detritivores are feeding on that organic matter and acquire it. So they are extremely connected, more than we might have thought in the past. Another aspect, especially in response to climate extreme, is that in the past, we tend to mostly think in terms of the trophic resource of the animals. But there is now some evidence that it is also the difference between the nematodes and the protists on the one hand. And the protists would be kind of not really animals, but it depends on the traditional classification there. But it's really kind of, it's a continuum really from, this is the point I want to make, from the microbes to the protists, the nematodes and the macro and the microarthropods, and eventually also the macroarthropods. It's a continuum, they're all connected. But in terms of basic physiology, there is a neat difference between the arthropods and the nematodes. And this is because nematodes basically need the water. Mostly, yes, they live in the soil, but they actually live in the water of the soil, or very strictly associated with the water. And the arthropods actually live more in the space where there is air. They need air. And so that's a very major difference between the two. And we are observing that because of that, most likely, they respond differently to drought and flood. And so certainly they are very useful in terms of indicating changes, but we don't know so much in terms of the overall functional implications. And now it plays out when they interact with the microbes in the plants. Yeah, so that's what I wanted to say about that.
So I could imagine from a food web perspective that if the higher trophic levels are responding and recovering in a different way after extreme events, then that gives all kinds of instabilities to the system, which makes the resilience issue much more complex, because when do you get really have a recovery of the original state, or something which is functionally the same as the original state?
Well, at the theoretical level, the microarthropods and the macroarthropods, and also the nematode, the predaceous nematodes, are coupling all the microbial energy channels. And this is central to the stability of the food web. At least this is what theory says. And so definitely, yeah, what you say is definitely very important. But surprisingly, there is no so much experimental evidence about that, especially with manipulations that involve things like spell of drought, maybe in alternation with other kind of perturbations.
Yeah, so there's a recent paper of Matti Thakur. He tried to work on that from so to increase the complexity in these trophic change, and then see how extreme drought, for example, was just hitting the different components in a different way. So I think that's one of the steps that would be really interesting to follow. So I see that we still have about a quarter left, a bit less. One of the aspects that I would like to discuss in the end is, so what is our message to a wider audience, or to policymakers? So what can we tell now? And I'm just now going to open it up for the one who wants to jump in first. If we would have to make 2 minutes at a bus stop or an elevator pitch to a politician, for example, or to a manager, what do we say?
Well, I can start by saying that we need to incorporate soil biodiversity in our conservation efforts. Because actually what is going on below ground is very important for above ground. And I guess sort of people are maybe starting to realize that that's the case in, like, you know, agricultural systems. We need a healthy soil. But I think it's important that people also realize that it really matters what is going on below ground, for actually how the system not only functions under sort of normal conditions, but also in response to these, these future climate events, these extreme climatic events. And in particular, as I said in one of the earlier questions, I think we need to sort of see this in interaction with the above ground world.
So what are the experimental results that you would show them to give evidence of your statement?
You're asking me?
Yes.
Well, there are quite a few, actually. Well, first of all, I would show them Cameron's paper about the multifunctionality. That's not in response to disturbance, obviously, but it's a very nice example to show that soil biodiversity does actually matter. And as actually some critics have pointed out to me, is that most studies of the benefit of soil biodiversity for ecosystem functioning, and in particular in agricultural systems, are correlative, because it's very, very difficult to manipulate these systems and to manipulate soil biodiversity in a relevant real world context. So many of these studies are correlative. So they show that management, for example, impacts soil biodiversity, and that this results in a system that responds, for example, differently to disturbances. But I think we need more of that experimental evidence. Obviously, I would also show them a study that I did in 2012 that shows that these different management systems with very different soil food webs respond differently to drought, and that it's actually the sort of the extensive system with the more fungal dominated soil food web that continues to function better under these disturbances. So that's one of the things I would show them. Yeah.
Good. I also show your paper always if I have to answer this question. The 2012.
Oh, it's still good, I think.
Yes. So other points of view on how to spread this news to a wider audience, or to policymakers or managers. Cameron, Mary, Tancredi.
So I'll comment on that, because we were just having a discussion at the end of a workshop very recently. And clearly you start with the importance of the soil and its biotic functionality, the AMF and the nitrogen fixation and the like, in crop production. But to convince our policymakers, I think we really have to move beyond simply agriculture, even though it's really important, and start to speak to the response of the entire terrestrial system as it is impacted by the organisms in the soil, particularly in the conversion of plant root carbon into stabilized soil organic carbon. And as it changes, particularly the dry down, wet up dynamics change, the pulses of CO2 and N2O that come from soil, both of which are critically important in climate change, global warming, N2O, of course, in ozone. And so I think the expectation is, yes, soil is important for agricultural productivity, but I do think we need to take a couple of giant steps beyond that, at least in this country, in order to convince the policymakers.
Yeah, if I look on the internet and I see this graph of costs involved in increasing extreme events, then I think that's something which could convince some people, especially if we would have some answer of how to reduce those costs. Well, for the ones who just only look through their wallet, of course. Cameron.
Yeah, so this is a really obviously relevant and important question for me working for the federal government in agriculture here. So there is quite some concern about soil biodiversity, and there's some growing awareness that soil biodiversity is quite important for sustainability. So some of the farmers, they're concerned about the productivity of their lands, especially under climate change events. And they hear all the time that and feel and experience warmer, drier summers. And so what can they do? So that, some of the policymakers in certain places in Canada and in Switzerland have sort of these guidelines that you should have at least a 3-year rotation and incorporate a lay year and things like that. So it's coming, but it's a lot of work and a lot of research, as we all know, to study soil biodiversity and its critical role in ecosystem resilience and buffering against climate. And if we see some long-term studies where soil organic carbon is being lost from some of these cropping systems, we need to find better ways to manage that and really demonstrate that it's working to promote soil biodiversity. So for example, things like regenerative agriculture, I feel has quite a bit of potential in promoting soil biodiversity and to help buffer against some of these climate extremes. So it's there. I think the awareness is growing, but often the question is, where's the evidence to convince me? I think, like you mentioned, what graph would you show? And yeah, the money or the subsidies, and to do the research and for it to be implemented in a real world scenario, I think is where we need to problem solve the most.
Okay, and Tancredi.
I think the others already said the most important things, really. What I will add is a warning to the policymakers, and that is that it takes hundreds of years, maybe thousands of years, to develop soil and its biodiversity. It takes the blink of an eye to lose it. And this is general, not just agricultural land, tropical forest, everything. And so we want to prevent this rather than try to respond to this.
I'm just looking at, so I see Lilia Serrano just asks, what happens with soil microbial resilience when there are no plants? So can we say something about the desert type of soils? Are they responding in the same way, or is just making the plant very different from a plantless system? Who has a suggestion there?
Well, I can say something, because actually, generally, when people look at how different plants sort of moderate below ground response to drought, well, quite often they don't actually include a soil without plants. So what actually just some very recent results that that my postdoc Alex Williams just shared with me just before this presentation is that because we imposed soil and a couple of different plant species to drought and warming, like an actual heat wave. And it's very, very clear that the soil enzymes and the soil carbon profile, metabolomic profile, respond much, much stronger to these disturbances when there are no plants. So you very, very clearly see that soil respond, but when you have the plant species growing, the effect of these different treatments is much more subtle. So it seems actually that in this experiments, the plants sort of buffer the impact of these disturbances.
Okay.
But we're yet to look at the microbial communities actually, but looking at the enzymes and the carbon profiles, that's what it very clearly shows.
So I just see that we have 4 minutes left. Do we want to give a message to the ones looking at soil who don't look at soil biodiversity? There are many.
So in terms of like soil scientists who focus on like carbon specifically, or?
Well, in this case, it's related to extreme events. So if you are interested in looking at extreme events from a soil perspective without looking at soil biodiversity, what are you missing then?
I would say the whole engine that's driving the whole thing. So, you know, the carbon cycle, the nitrogen cycle, it's pretty hard to consider these aspects of the soil without considering soil biodiversity and soil microbes and meso and mesofauna that drive this whole system and sort of keep the whole system intact. So it's all sort of one big living organism, and the abiotic factors are sort of a part of that as well, I would say. Soil organic matter is also a good indicator of soil health and soil carbon and all those aspects as well. I'm not sure if there's others who want to jump in quick.
I like this idea that you are looking at a car without an engine. I see the others looking puzzled.
I would say that I show my last slide was soil water, one of the simplest thing, and it's very easy to measure, and then, you know, soil scientists do measure that. But surely the velocity at which soil water recover and the velocity at which the system can recover will actually depend also on the life you have in the soil. So you can't just ignore it. Ignoring it may mean missing an opportunity to improve your ability to restore the system or to improve your ability to make the system more resistant to perturbations.
Okay. Thank you very much. I think that I'm going to round off. It's almost 2 minutes to the half hour. And what the half hour is depends on where you are. So I would like to thank the speakers very much. I really enjoyed the presentations, the introductions. They generated great questions. Of course, we could not address every question in detail. I've tried to take some of the combined issues from the questions and then present them. I hope they can be kept in the Q&A. I also would like to thank Diana Wall and her team, Monica and Juliet, for organizing this. And this series is running so well that it's going to be continued. And the next one, we are thinking about that. We also got the FAO Soil Biodiversity meeting in April. So keep an eye on the Global Soil Biodiversity Initiative website. And if you have an idea yourself for a webinar, just mention it to Diana and her team. And yeah, for the rest, I want to wish everybody a good afternoon, good evening, good morning, and see you next time at the GSBI. Thank you very much. Bye-bye.